Our next presentation pivots from the pediatrics department to civil and environmental engineering. Laura Kwong is a PhD candidate, and she's going to talk about ingestion of soil and food contribute more to children's intake of fecal matter than ingestion of water. So I think we're all excited before lunch to hear about ingesting fecal matter. Over to you, Laura.
Hopefully my results are a little bit more site-specific and that we are doing a little bit better on our food hygiene here at Stanford. So yes, as Steve mentioned, I'm in civil and environmental engineering and lucky to work with Steve Luby and Jenna Davis for my dissertation work. I am very interested in fecal transmission pathways. How are children exposed to the feces that can result in diarrhea? As you may know, there's still about 500,000 children that die from diarrhea every year. So how does that happen? How are the feces getting from the environment into their mouths, right? We're talking about fecal oral pathogens, so we need to look at that entire loop. So feces can be transmitted through water, flies, soil, hands, through intermediate vehicles such as food and objects into a human, right? And there's many ways we can interrupt these pathways, including sanitation interventions, which have been shown to reduce diarrhea by 32%, as well as drinking water treatment and hand washing interventions. There are also some other interventions which have been less well studied in sort of randomized control trial in the intervention space. These include food hygiene and clean floors and clean play spaces. So when I first started at Stanford and I saw this diagram, I said, so which of these is the most important, right? Which of these pathways has the largest relative contribution to children's intake of fecal matter? Because that can help direct our interventions. What should we focus on when we're trying to stem diarrhea? Does a more accurate diagram actually look like this? In which case we should focus on drinking water treatment and hand washing? Or maybe the diagram looks more like this, and we should really focus on food hygiene and clean floors. In order to answer this question and direct our energies in the right place, we need to find out the relative contribution of these different pathways. To my surprise, there was very little literature on this, and no one I asked could answer my question about which of these pathways was the largest relative contributor. There was one study in Tanzania which compared just two pathways, water and hands, and found that 97% of the fecal matter came through children putting their hands in their mouth, right, rather than drinking water. My question was, how would this change if you included the other pathways in your research? So we took this research to Bangladesh, where it was an add-on study to the water, sanitation, hygiene, and nutrition trial called WASH Benefits, run by Steve Luby. And we tried to gather location-specific data to answer this question. So we needed data on environmental contamination, right? What is the level of contamination in these different environmental media? How are children contacting that environment and interacting with it? And sort of what are the transfer efficiencies of contamination in the environment into a child, right? And we wanted all of this data to be as location-specific as possible. So first, we collected environmental samples. We looked at pond water, stored water, drinking water. We collected flies from outside a kitchen area, soil from the area in the courtyard where the child last played, children hand rinse samples, food that they were about to be fed, and objects. And because we can't test all the objects in the house, we used a plastic ball that was rolling around the house for 24 hours. We collected over 10,000 samples in both the rainy and the dry seasons. And you can see here some of our sample collection. We took these samples to the lab where they were tested by IDEXX for fecal coliforms and E. coli. E. coli is an indicator of fecal contamination, right? So we're not looking at E. coli as the pathogen itself, which is giving children diarrhea, but rather just an indication that that sample was contaminated with fecal matter, which could, in some cases, could have a pathogen in it. So the results you can see here, that about 100% of soil and ponds were contaminated with E. coli at extremely high levels. So the soil was contaminated with approximately 125,000 E. coli per gram, right? And this is partially because there are chickens and cows and goats running around. There's also inadequate fecal sludge management from human waste. And so soil is extremely contaminated, and the ponds are also. But this doesn't answer the question of which pathway is the most important, right? Because children don't go swimming a lot when they're under 2 years old. Unfortunately, in Bangladesh, sometimes they try, and drowning is a very high cause of death among children under 5 in Bangladesh. But we needed to find more about the interaction of children with these environmental media to understand the relative contribution. In order to do that, we conducted observations of children at their homes. We conducted 84 structured observations, 150 structured observations, and 84 video observations of children just being kids and recording everything they did. So you can see here a clip from one of our videos. So I'll show you how we recorded this data. This child in the darker clothing is playing outside, and her hand is constantly contacting concrete, right? We also record that nobody else is interacting with this child or placing anything in their mouth. And the result is an Excel file that has detailed information, second-by-second information, on how this child is interacting with the environment. The result is the frequency per hour of the things that are going into the child's mouth. As many of you with children know, children are very frequently putting everything into their mouths, right? So as you can see, the rate that they put their hands in their mouth when they're not eating decreases as they age, as does the frequency with which they're placing objects in their mouth. But the frequency that they're placing hands in their mouth when they're eating goes up. And this is because in Bangladesh, it's customary to eat with your hands rather than utensils, so it makes sense that as children are growing, they're going to put their hands in their mouth more to eat. Right? We were also specifically interested in their ingestion of soil. Soil has been suggested to be, ingestion of soil has been suggested to be important for hindering child development. And so we wanted to compare this to other studies that have looked at the prevalence of soil ingestion. Here you can see our results in the dark red compared to studies that were conducted both in Bangladesh and Zimbabwe. Kids eat dirt, right? I remember making a mud pie, maybe you do too, and sometimes it's just interesting to see what that tastes like. So kids that were 6 to 24 months old, about a third of them were ingesting some amount of soil. And this is direct ingestion, not crawling and then you just incidentally put your hand in your mouth, but really just like, I wonder how this tastes, right? We were also interested in direct ingestion of feces, as this has been suggested as an important pathway for total fecal intake, right? And we did not notice fecal ingestion. I will say from watching hundreds of hours of video that there was one child who went over to a pile of cooking fuel, which happens to be cow dung in many places in rural Bangladesh, and put that into their mouths. But the mother came over immediately and took it out. So putting all of this information together with the transfer efficiencies, the transfer efficiencies tell us how much of the contamination in that media will go into the child. It's not always 100%. For things that are directly ingested, it would be 100%, but things that are less directly and have to go through another source, we're thinking it more at 66%. We put this information together to better understand the entire set of fecal transmission pathways. And the way that we do this is using a Monte-Carlo simulation. So we have, for example, if we're looking at drinking water, we have a distribution for the concentration of contamination in that drinking water. We also have a distribution, this is hypothetical, which represents their ingestion of drinking water and the transfer efficiency. We could use a deterministic model, which just takes maybe the median or the mean value and combines it to get an estimate of their exposure. But instead, we used a probabilistic model, which compares, which draws from a different distribution every time, do many iterations, and we can come up with an entire distribution of their exposure. So we do this for all of the sources that we are interested in, and the result is here. You can see on the x-axis is the type of media that they're exposed to, and the y-axis is the total percent of the contribution of fecal matter they're ingesting. And you can see for different children of different age groups, there are differences. For children in the youngest age group, placing objects in their mouth is a large contributor. For children in 6 to 24 months old, placing soil in their mouth was much more important. And for older children, consuming food was important. So this suggests that a different perspective of looking at children has identified previously neglected pathways, and maybe indicates why some WASH interventions have not been quite as successful as we expected, because we weren't targeting those primary pathways. It also indicates that the relative importance of these different pathways differs by age. So we really need to consider the age of the child when we're thinking about the appropriate intervention. Furthermore, it suggests that we really need more development and evaluation of techniques that could address children's contact with contaminated soil, food, and objects. We're identified as those main pathways of fecal transmission. Happy to take any questions you might have.
Great talk, thank you. I was wondering if you're going to tease apart the human and the animal fecal matter, or if you think they're both equally important in the transmission of, fecal oral diseases.
Yeah, great question. So we published 2 papers actually on the relative contamination with human versus animal feces, and that was determined by doing source tracking, using source tracking markers. And the contribution of animal feces is extremely high, mostly from ruminants. It's a little bit difficult to determine what is from animals versus humans because they live in such close quarters that there's a lot of crossing. And the fecal source tracking markers that are used to distinguish animals and humans in the US are not as sensitive and specific as in Bangladesh. So there's definitely some human matter we know from other ways of studying, but the fecal source tracking shows that it's mostly animals. Yeah.
So children who grow up in environments where we've minimized their contact with soil and fecal matter as much as possible, say here in Palo Alto, still rapidly acquire a gut microbiome, which is bacteria we think they acquire from human populations. What do you think the transmission rates for that process are in these environments that are very clean?
Yeah, that's a great question. So continuation of, building on this study, we're doing a study in Fiji and Indonesia, which we hope to look at very similar lines of fecal transmission. And just from visiting there, it looks like their transmission from soil might be a lot lower because their homes actually have floors that are not made out of dirt. They're made out of tile or concrete. And so that will help us better understand what the transmission looks like when you take out soil. And then if you looked at kids in the United States, if you would take out soil, food hygiene is much better. We have refrigerators, so contamination doesn't go as quickly, so you might really eliminate that pathway. I think that objects and hands would be the highest ones here. Looking at my cousins taking care of their kids and incessantly cleaning things with wipes, right? But it's not always going to stay clean, right? You have the ball rolling around in the backyard and it gets dirty before you clean it off the wipe, it goes into the mouth. So I would hypothesize that those are more important pathways here in the US. Any other questions? Yeah.
When everybody is exposed to basically everything at all times, constantly, 24 hours a day, do they have a more uniform microbiome than, let's say, in a country here where, if you grow up super clean or, that's probably the next research project, right?
Yeah, well, actually, I have good friends. Jess Grenby in the back corner over there is looking at the gut microbiome based on these interventions and how that differs between the control and intervention groups. And Liz Costello and Jess also just got funded by a CAGH seed grant to look more at children and their gut microbiome and how it relates to their growth over 2 years. So I guess those results are upcoming. Thank you.